Hello and welcome to the ATG podcast. Today we're talking with Richard Jefferson. Richard was originally a plant geneticist, but perhaps he's best known in the scholarly community for setting up the Lens, a corpus of patents, patent sequences, and scholarly content. So in this interview, Richard will talk about his background and how he moved from biology to advocate of open science and his role as a social entrepreneur. So welcome, Richard, and thanks for joining.
Thank you very much, Michael.
So let's start with your background. I believe you grew up in California.
Well, insofar as anyone in California grows up, but yes, I certainly aged in California in the early stages of my life. Yes, I was born in Santa Cruz, California, and then raised in Silicon Valley when it was orchards, and that should give some dating of the carbon. And did my undergraduate in University of California.
And how did you get interested in you studied science? How did you get interested in science?
Oh, I don't know if I've ever not been interested in science. It's not something I ever felt I had to work at. It was just like oxygen. So my mother was a professional librarian, and I was inundated with evidence-based thinking from my first breath, pretty much. And so when I started hanging around with people who did science, my brother was a physicist, elder brother, and it was natural. So it's not like something I decided I have to work at this. It was like unavoidable.
But you focused, you hit science at a particularly fertile time for biology because you studied genetics.
Well, more than that, actually, I was really talk about good luck. I started my undergraduate at the University of California at Santa Barbara in the year that recombinant DNA was invented. And my first exposure within a couple of weeks of starting as an undergraduate was it was really interesting. There was and this actually does set the tone for much of what I think you'd like to hear more about. It was in the lab of a man named John Carbon, who had just literally 2 weeks away earlier had returned from his sabbatical with Paul Berg, a Nobel laureate, shortly thereafter, for inventing recombinant DNA with some colleagues at San Francisco and Stanford. And so there was I, wet behind the ears undergraduate, just absolutely entranced with biology and hearing about this harbinger of a revolution in biology based on DNA and molecular biology. And I was so excited about this that I engaged with John Carbon, and he did something he'd never done. He brought an 18-year-old undergraduate into his laboratory to learn the craft of in the presence of the real rock stars, or the soon-to-be rock stars. It was an incredibly exciting time. And that pretty much, cast the die for me.
Sounds amazing. And I believe you even got to spend a year in Edinburgh during your undergraduate degree. How did that happen?
Well, okay, recombinant DNA, when I got engaged in it when I was 18, that was 1974, I embedded myself in the literature. So I was probably one of the few people who looked at some of the major journals as effectively as narratives from cover to cover. I didn't fully grok that, you pick and choose in a journal and you read a paper that's interesting to you. So I was in the lab washing dishes, cleaning up, pouring Petri dishes, and also getting to do some of my own research as an 18-year-old. It was incredible. But the journals, JBACT was very important Journal of Bacteriology back then from the American Society of Microbiology, PNAS, a number of journals like that, Proceedings of the National Academy. And they would arrive in the lab, there were 8 postdoctoral fellows just absolutely going hammer and tongs at this whole new field. And they'd arrive, and I just sort of crease the beginning, and I just start reading it as if it was a novel, chapter 1, chapter 2. They were actually papers. But it was, it was so captivating. It was beyond riveting. Seduction was the word, Michael. It actually seduced me into realizing that the enormous potential of methodology. I was watching everything change. So in the course of the first 2 years, I became obsessed with the premise of methodology being a key to my personality. The idea of figuring things out didn't excite me in science. Almost all scientists love to figure things out. I like to figure out how to figure them out. So from that point of view, developing a methodology is extraordinarily exciting. And back then, contrary perhaps to the thoughts of some of the open science advocates, now science wasn't broken. It was incredibly communicative. Now, we would be on the phone all the time with the other competing labs who are our colleagues. And of course, as the young Turk, I got to answer the phone all the time. Hey, Bo, it's for you. and getting to talk to everybody from Paul Berg to Paul Schimmel. They weren't all named Paul, but that was the start of it. And saying so-and-so's got expression in yeast with that new vector. And it's just always very exciting. And it was absolutely open. When we had a new result, we'd be on the phone to Ron Davis at Stanford and so on and so forth. And it convinced me, without even having to study the process, that really science was about this aggregative, joyful development of new territory, intellectual territory, but also territory for humankind to improve or not. It was seductive beyond words. But to come to Edinburgh, at that time, I was, 2 whole years at University of California, working in the lab 10 hours a day. And I thought, there's got to be more to life than this. And I've always been deeply fond of Scots culture and, strangely enough, Scottish music, and all of that. And for some reason, I immediately realized in reading all these journals that a number of them were coming out of 1 or 2 labs at University of Edinburgh in the Department of Molecular Biology down in King's Buildings. And it's from Ken and Noreen Murray, 2 different, they're married, but they ran different labs with different foci. Ken was the chair of the Department of Molecular Biology at that time. And he and my supervisor, mentor, talked to each other some regularity, because Ken was really the leader. Ken and Noreen both were leaders of the nascent molecular genetics world in Europe. And in particular, molecular biology evolved out of the complex logic of molecular genetics, which are not the same thing. If you can put it in a blender, it's molecular biology. If you have to actually look at it while it's alive, it might be molecular genetics. And Edinburgh was spectacular. I would say that of the 2 or 3 places in the world at that time that had that level of intellectual engagement and a sense of joy and creativity, there might have been certainly Jacob and Monod in Paris at the Pasteur, and 1 or 2 other groups. But Ken and Noreen's were great, and it was in Scotland, which I'd always wanted to go to. So University of California had an education abroad program, and I applied for it and was rejected. If it wasn't for music classes, I wouldn't have gotten a very good grade point average, because I spent all my time in the lab. So I wrote to Ken, and not understanding yet the British premise that the head of department, the chair, is not Ken, he's Professor Murray. And so I wrote to Ken and I said, "Hey, Ken," and copied it to him. "Hey, Noreen, I love your stuff. And John has been reading all this stuff about phage packaging, blah, 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 blah." And he was, being written to by this undergraduate? What's this all about? So he popped it over to whatever part of the university dealt with this and said, "What's all this about?" Ultimately, they recanted under duress and accepted me into this education abroad program, which was just a blast to be embedded with some of the brightest, most stellar people. Some of the great techniques that were being developed right then were being developed right there or in the MRC laboratory across the street from where I was working. So it was intoxicating, Michael. It was something where every fiber of my being was excited by how thrilling it was socially, intellectually, and emotionally. It's an incredibly great feeling to see, as a new technology or new technique develops, within days, weeks, months, through the telephone, of course, back then, it would be all over. And the way it would change the way people ask questions, how they answered them, whether they answered them, became a thrill to me. So I was addicted early on. And that's why Edinburgh, and I had a grand time playing music, riding bicycles, everything you do when you're an undergraduate, but also working 12 hours a day in the lab. It was a blast.
Weren't you overawed by being surrounded by people who were sort of several years older than you, with a lot more sort of education sort of behind them?
No, not even vaguely. Why would I be intimidated? That's exactly why I was there. I mean, if I wasn't there to tap into their craft, it would have been different. But and to be honest, in spite of the reputation that at that time was foolishly propagated that the British people, and Scots for that matter, are reserved, I found that there was a charming thrill that seems to be expressed when someone is wanted. So when I was going there saying, "I'm dying to pick your brains about this, how does this work?" I never got anything except a peer-to-peer response. I never said, "Oh, you don't know what you're talking about, Jefferson." Of course I didn't. That's why I'm talking to you. And it was a blast. I had a wonderful, wonderful time in Edinburgh. I'm just talking to you about it gets me all thrilled, because I haven't thought these thoughts for a while.
Sounds great. It's a lovely city. So you talked about a methodology, and the particular methodology that you came up with was this thing called the GOS reporter system. Tell us a bit about
Mhm.
that.
Well, I call it Gus, which sounds rather like a garage mechanic in Alberta, but it's a short term for glucuronidase. And it's a system I developed when I went on to graduate school after my Edinburgh time, which was as an undergraduate. I went to graduate school, and the place I chose wasn't the usual Harvard or MIT, but Boulder, Colorado, because my mentor, Carbon, said that, okay, you're a good scientist, very good scientist, but the thing you need to do is learn what questions to ask. And the people at Boulder, and this is again formative, were so collegial. So the United States has this strange phenomenon, sort of, it's like an athletics draft or a basketball draft. The top 10 or 12 candidates in graduate school, they apply to the same places, Harvard, MIT, Stanford, San Francisco, Berkeley, blah, 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 blah. And most of the same ones get into each of those. And then the different graduate schools will fly you out there to sort of bring you into their team, because often they think that dynamic undergraduates end up becoming dynamic graduate students, which means that they get an awful lot of good science out of these people who are foolish enough to think that it's fun to work 18 hours a day. So, and it is, or was, not so much now. So what happened is I applied grudgingly to Boulder because John Carbon had said that I should do that. And I got into all the usual places, of course, but also Boulder. And they flew me out there, and it was almost impossible to describe how it was building on all this other approach that I was taking, because I would notice that the first morning seeing the university or that graduate classes, they were all taking an advanced seminar class, but all of the faculty, the professors were in there with the graduate students. It was the most collegial environment. For the next, 6.5 years of my life, I was embedded in a place where the professors and the graduate students were in a seamless communication in many ways. It wasn't always perfect, but it was spectacularly interesting and informative. During that time, I landed, I'd love to fill you in on all the details about I seem to be like Zelig of molecular biology, I kid you not, where wherever I go, as soon as I leave, the person gets a Nobel Prize. It's really irritating. I think, can I just stay somewhere for a while? Oh, no, we need you to leave so I can get a Nobel Prize. So the first lab I landed in was this phenomenally lovely scientist named Tom Cech, C-E-C-H. And the work he was doing while I was fumbling around as a first-year graduate student ended up being part of the fabric that he won a Nobel Prize quite justifiably for, just like earlier on with John Carbon and with Paul Berg and stuff. So anyway, Boulder was a great place. I learned how to think about what the problem space is, not in terms of what can I ask, but rather the opposite, Michael, is what can't we ask? In other words, what is hard? And it's usually not hard because it's intellectually hard. It's hard because we don't have the tools. And this was grist to my mill. I just adored the idea that we could start rethinking about biology by saying, where are the bottlenecks in solving problems? So at that time, I was working on a small nematode, an animal millimeter long, called Caenorhabditis elegans, in a couple of labs that were, again, filled with Nobel laureates who were coming over from Cambridge. John Sulston was coming over. He's well known, and John White, and all these guys. And they kept getting Nobel Prizes all the time. It's just really irritating. But we were trying to figure out how a cell went from being, let's say, just a precursor cell in an embryo to suddenly one of the daughter cells becomes a nerve cell, the other one becomes a muscle cell. How do it know? And we figured out that we needed a tool to ask questions experimentally in real time in living cells to say, what's going through your mind, Pickles? I mean, what are you thinking about when something changes? I mean, and we needed an experimental tool. So I decided to develop what's called a reporter gene system. That would be a system in which if you could do transgenesis, and parenthetically, no one had ever genetically engineered an animal before, I never really viewed those things as in the way. They're just, okay, that's what we got to do to get this done. And to build something where you'd make compounds that would turn a beautiful fluorescent color under illumination during the living development of the egg of this worm. So we could actually then do experimental biology and say, well, does it have to be next to this cell to know that it's just changed? All those sorts of questions can only be asked if you have a tool that lets you resolve it. It's rather a heuristic of sorts, Michael. So if you have a way of measuring or experiencing something, you have a way of optimizing it. But until you do, you're in the dark. And so I developed Gus for that. It's too long a story to recount in this interview, but basically because of, again, conflation or congruence of enormously generous people. I bashed my head against the wall trying to figure out how to do this, and someone in another lab upstairs would say, well, have you considered such and such? Science was not broken. It was exciting. You need to get better, as you'll see, but it wasn't a broken science. It was fantastic. And so I developed this tool that allowed us to genetically engineer the C. elegans, and then to visualize in the living cell when a change happened, when it knew it was different than it was a few minutes ago. It was very powerful. But in the course of that, I got absolutely burnt out because methodology development in science is very secondary to solving a problem associated with one's prestige. And I of course felt exactly the opposite. I thought it's incredibly easy to ask and answer questions if the tool lets you do so. But if you lack that tool, and this is very true of all the other things you and I are going to be talking about, if you lack a particular tool, you're constrained in the sorts of things you can ask, the sorts of problems you can solve. So for my personality, coming from a performing arts family and a librarian, the idea of crafting tools and developing them and sharing them was seductively beautiful. The idea that you hoard things, it can be possible for the kind of science that is about discovery. It's about ego and discovery. I have to discover how this so I can get the credit for it. With a tool, if you don't share it, nobody knows you've done it. And it's the most absolutely selfish act on earth because it feels so good when people use the tool you've developed. It's really cool. So I developed that, but in the course of that, I realized that developmental biology of animals is enormously different than that which I was excited by, which was how does the environment influence the behavior of complex systems? So I became obsessed long before I was exposed to it to plant and environmental biology and agriculture. And so I set out to get a postdoc, a self-funded postdoc, to go work on developing these same types of tools to ask questions about plants. And I thought crop plants would be exciting because there's such a body of knowledge about crop plants that may not exist for any particular non-crop plant, with a few exceptions. So I was applying to work in Cambridge to postdoc at what was then called the Plant Breeding Institute in Trumpington. Because again, there were only a few places in the world that had world-class molecular genetics, as well as plant breeding, agronomy, all of the different crafts you need to understand what happens in the field. So I decided I would, pull up stumps and take my my tool that I developed for the worm, the Caenorhabditis, and try to move to Cambridge. But it took me 2 years to get a postdoc fellowship to go there because everybody wanted to fund those people who solved a fundamental question, irrespective of you can't solve it if the tool isn't there. So I proposed to develop a tool. And this is another point that's relevant to our discussion. I applied for postdocs, and every single one of them was opaque, meaning the determination of why I wasn't getting it was absolutely opaque. So it was soul crushing to apply, and I knew I was better than anybody else. And all my friends were getting these postdocs from the NSF or from the Helen Hay Whitney or the Damon Runyon or the American Cancer Society, real prestigious ones, to go to these other places. And I was just, am I supposed to turn out the lights when I leave as the last guy? It was really, really terrifyingly frustrating because I was so tired and burnt out from having developed this technology. Everybody says, oh, you won't work. It worked. So finally, the last possible postdoc fellowship, the reviews came in. And the last one that I had applied for, thinking it was no chance, because it was the NIH, and it was to work not in the United States, but in the UK. It was not to work on medicine or health. It was to work on the humble potato. It was to work on molecular biology of crop plants. So it was working for someone who's roughly my age, working in a lab with someone who's roughly my age. So it was a young guy, really, really nice man, who was the nominal group leader, a Kiwi, there in Cambridge. And so I was applying for all the wrong things at NIH, but the difference was this. The NIH shared the reviews. This is going to be very resonant to the librarian community, those that are pushing certain of the open access paradigms. They let me read the reviews. They couldn't keep them secret. And I remember reading them, and one of them saying something very, very, very extremely flattering. If this happens, it'll be a major breakthrough in plant sciences and in agriculture, in spite of his recommendation. And I thought to myself, what? It turns out that at least one of the people that was writing me a recommendation, I'm not going to name names, had basically found my personality a little bit over the top and didn't hesitate to mention that to the reviewers. But that worked okay in the old boys club, and it worked just great in the old boys club with the American Cancer Society, Damon Runyon, Helen Whitney, all the other ones, and NSF at the time. But NIH, they weighed risk and reward, and they said, well, look at this, what he's done already. He may be a pain in the neck and talk too fast, but wow, what a project if he can pull it off. And they thought to themselves, what's our risk? He's the one who's going to work his ass off on this. If it works, the upside is going to be really good. And they believed in me. And I just remember, it sounds a little emotional, but I actually just broke down and completely broke down for probably for days. I don't even remember that some panel somewhere believed in me and expressed that in a way that I could read because nobody else had. So then I took my NIH postdoc and went to Cambridge and lived, amongst other places, on the same street where you live, though it sounds like a musical, on the street where you live, Michael. Anyway, and I, again, had a marvelous time doing something else that was relevant to the audience you're supposed to talk to. That is, I discovered that the cloisters, the guild of professional science, has sub-guilds, and you don't really want to transfer out of those because there's a huge psychological and sometimes financial, but certainly prestige or rent you extract from being in an elite guild, whether you're the molecular biologist or the physiologist or the physicist or what have you. And your approval is based on performance within a guild, a set of norms that are associated with your behavior and your promotion. Oh my God, he's published in these journals, whatever else. And those norms were very well entrenched within sub-guilds and in the general guild of experimental or theoretical sciences. And what I found was exciting was, and really exciting and invigorating, was when I fully realized, embraced the fact that this isn't about me or science. Plant biology is not a joke. It's not just for fun. There are billions of people whose livelihood, well, all of us, one way or another, depend on successful agriculture. And as a species, as a set of societies, we depend on it being done sustainably. And I looked around, there were people who knew lots about agriculture, but they weren't like me. They weren't molecular biologists. They were the agronomists. They worked with soil, with farmers, God forbid, and with the crops themselves. And they had a wealth of firsthand knowledge of what the problems were, what the impediments to doing things well were. Were they being brought into the definition of the problem set? No, of course not. They had a completely different set of norms. The guilds of plant breeding, they were associated with outcomes, always outcomes. It's the one O, not open, but outcomes that really started to get under my skin, realizing that most of the postdocs and grad students I was working with were from what were then called the developing world. So it would be Eastern Europe at the time, Latin America, Asia, Africa, all over. And these were my friends. These are people I was working with, many of them actually older than me, but having their one bite of the cherry to actually publish a paper while coming out of Cambridge. So I started getting kind of angry about things because what seemed to me was that there was this entrenched old boys club, and it was unfortunately mostly boys or men, where the students and postdocs, generally from disadvantaged economies and some places where this was a crucial piece of their education, they were treated poorly. They were used as lab rats or as cannon fodder or plant tissue culture fodder to do the grunt work, which advanced the careers of the rock stars. And I was just starting to get furious. And so I'd go to the other end of the institute, which to its great credit was one of the few that had serious cytologists, cell biologists, and agronomists, people with real muddy boots who didn't actually probably own non-muddy shoes, and plant breeders. And I started hanging out with them, and I discovered that there's a reason they're called salt of the earth. I mean, they hang out with the earth, and they talk to the people who turn the earth and work the earth. And because of that, their efforts are always ground truthed by real people and real outcomes. And this was the kind of science I thought, this is so beautiful. They're listening to people and letting that inform what are the problems we should work on. I mean, wheat breeders don't just make a better wheat because it's better tasting for them. They canvass the farmers. What are the problems you're having growing it? They canvass the millers and the bakers, and they would listen to what the issues are with the atrocious soft wheats in the UK at the time and start breeding some brilliant wheats. Plant Breeding Institute was probably the home of the greatest British wheat breeders and sugar beet and potato, parenthetically. All of these people responded to imperatives from real human beings, working people who understood themselves empirically through and communicated through a really important science tool that most scientists still to this day don't appreciate, and that's narrative. Basically, when you get really complicated or, God forbid, complex systems, you reduce the way to describe it to very, very few. It's extremely, Complicated systems are describable laboriously in their component parts, but complex systems, those that have emergent properties, those that are intrinsically variable, if variables are part of the system, often it's narrative. It's somebody telling you a story, but telling somebody a story often in the not very articulate way that somebody who's really under time pressure to get their crop out, it may not sound like a story, but it is. And these people, who I admired enormously, were really good at going down to the unicorn on Trumpington, oh God, where was it? Just near the PBI. And they would sit down with the local farmers and have a pint or two or more. And they would listen and help tease out the narrative of what the issues were. And I just thought this, oh my God, this is what's missing in science, is listening to narratives and communicating back in narratives. Because some of the minutiae of genetics and plant breeding or all that, they were not necessarily in the lexicon of a potato farmer from East Anglia. But that said, they knew a lot about potato farming, the economics, the biology. So it was a wonderful reciprocal development process. But in molecular genetics, where I was a young rock star by that time, because this technique I'd invented was used by me and my colleagues to develop the first transgenic food crop, which was potatoes. Very, very pedestrian, but they were potatoes and they were beautiful. And we had one beautifully illegal dinner, but that's another story for another interview. The first transgenic dinner, we had transgenic potatoes au gratin, all different ways of making it. And look at me. That's what comes. Anyway, so to come back to it, the narrative, the craft of giving and receiving narrative was dealing intrinsically with complex systems, which were almost anathema to the molecular geneticist, including the frustrated molecular geneticist and me. We had developed transgenics for the first time. And what we set out to do is not to fix the potato. The first field trial we did, which was there in Trumpington, and this is really funny. By accident, I found a year later, we beat Monsanto by 1 day to being the first field trial of a transgenic food crop. Monsanto did it in Jerseyville, Illinois on June 2nd, 1987, I think. And we did it June 1st. We didn't know that at the time. It was a pure accident. And ours was done with the son of a local farmer driving the tractor and a whole bunch of the local folks planting it, no moon suits. And we didn't plant it to make Roundup ready or something like that. We planted it with thousands and thousands of plants to ask a question. How do genes behave in the real world when they're in soil, when the light is variable, when there's pests, when there's stuff? How do genes behave? So we used this tool, GUS, which made the pretty colors of the fluorescence when it was expressing, and we did thousands and thousands of analyses. And the take-home message, of course, was genes are going all over the place. The activity within a gene in what looks like a sessile, boring plant was just crazy exciting. It was doing all sorts of things because it can't run away. It was listening to the temperature, the humidity, the shading, that's coming in, the microbes coming in. So I realized then that our tools were, it was a big experiment and no one was going to repeat it because it was too hard. So I said, our tools are wrong. Our tools are wrong on two fronts. One is social and the other is technical. And those are intertwined, because if you don't design the tools for a particular hand, they won't be useful to the particular person. So at that time, and this is again something that will be resonant to the open aficionados, I was getting so frustrated at this guild separation of guilds. You didn't cross between guilds. You didn't talk to people who, God forbid, weren't even in the science super guild. I thought a huge lost opportunity, but it was worse than that. I actually got really, really angry because I was becoming very close to a lot of the grad students and postdocs and even young assistant professors who were on a first sabbatical of PBI. PBI was a great place. Their director at the time, Peter Day, ended up becoming a very close friend of mine. And they were an enormous resource that wasn't being harnessed well. And I think they know so much. They know about that, and they care so much. And that's a big thing. They cared and they knew. So what ended up happening is I'd watched these squandered, committed, capable people, because they might have an albedo that's different than mine. And for those whose vocabulary doesn't go there, that's just the reflectance. Okay, it's not. Anyway, so they were from a different place, and those places were fascinating, and they had stories. So I got mad. They seemed left out like this. So here's where the beginning of the whole open thing happened for me. Real open. I don't like to publish much. I find publishing very ad hominem, and the reviews can sometimes be a little spiteful, especially if you're really trying to be a little edgy. And I don't like to publish when it's not ready for prime time. I really want to get it out there so people can just use it. So I hadn't published the Gus stuff. We'd done the first transgenic, and people knew through the rumor mill that we'd done this, but we hadn't published it, so they could say, first, we're the first. What ended up happening is a professor, a very famous professor from a university in Western United States, very famous at the time, who was also something of a jerk. He was a big jerk. Okay, so it wasn't something of a jerk. I was being polite. If I used his name, I would just say he was less than palatable. But he wrote to my colleague, the guy I worked with, the principal investigator of this lab at the PEB. And he said, hey, I won't tell you his name because he wasn't a bad guy. He was a great guy, the guy, my local colleague. He said, I hear through the rumor mill you've developed this incredible new tool. What is it called? SUG, Gus, whatever it is. We haven't, could you send it to us? And you and I could do these experiments and we can get some papers in Cell, Nature, Science, you name it. And this is where the guild finally broke for me. I saw this letter pasted over this guy, my colleague's bench. And he was pipetting the 10 or 12 little tubes of DNAs I had constructed, promoter vectors that fused reading frames with this new reporter gene, all sequenced by me, all constructed by me, all used in transgenesis by me, by me, not by a bunch of people working for me. And he was filling them to go back with that. And I lost it. I mean, I'm not a person who handles anger or even expresses anger very well at all. But this time I just ripped it off the bench and I said, no way. He's getting it at the same time as everybody else on Earth. It's just no way. I just don't like the prestige mill for prior exit. And so I used my NIH money to buy about 1,000 bubble wrap mailing bags. And I spent a couple of months. I first wrote quickly, which is anyone who knows me knows, what, he wrote something? 40 or 50 page user's manual in great detail, the DNA sequences, everything else, and put about, between 10 and 20 tubes of DNA of different what are called reading frames and everything there, and including samples of the new compounds I made to measure this stuff and the techniques. And I made, hundreds and hundreds, maybe even 1,000 of these things. And then a friend of mine who was at University of Georgia, that's where there's a society that had a little A5 size magazine. You wouldn't call it a journal really, called Plant Molecular Biology Reporter. And it came out every quarter, and it was maybe 20 pages. And it wasn't gossip. It was, little papers that are submitted, but not really, it was zero prestige. But they had that community, if you want to call it that had a mailing list. And the mailing list was pretty much everyone on Earth that had cared about plant biotechnology for agriculture. So labs in Bangladesh, Berkeley, Botswana, any place that started with a B. And so they were all on the mailing list. And this was, of course, before email. So I called up my friend Wayne Parrott, who's just a remarkable scientist at the University of Georgia. I said, Wayne, can you get me a copy of this? He says, well, I don't know if I can. It's not public. I said, well, where is it? He says, it's over in Joel's lab. Well, can you get it? Well, yeah. Can you fax it? Yeah, because faxes were the thing, And so he faxed a list with mailing addresses for about 600 labs around the world, 600 labs, everybody that cared about this, because it would cost $5 to join this society or something, or get this journal. It wasn't, expensive. And so I sent it out to everybody with this thing. And this is before I'd published it, for sure, and certainly before I'd even patented it, which is coming to another thing, with the idea that I really wanted to see small local businesses built using science and biotechnology to improve the lot of their local communities, farmers and business people. Because it's not going to be done by the public sector. Much as everybody claims, public sector, it doesn't develop products. And to develop a product is hugely complicated, and you have to get all the moving pieces together and incentivized. And it doesn't matter if your science is great if you don't have marketing. It doesn't matter if the science is great if you don't have regulation or intellectual property rights. You got to get them all. So I sent it out to everybody naively thinking, well, this is going to open things up. So this might have been the birth of open science in its modern form, sending everything out long before it was published, with full disclosure of all the DNA sequences, equivalent to software these days, and the materials itself, even unbidden. It wasn't like people wrote to me and said, can I have this? Because they didn't know I existed. Almost nobody did. And it landed on everybody's desk, everybody's. And some of them were really well prepared to use it. So it landed on some friends of mine's desk that were in St. Louis, people by that time I knew real well. and in Middleton, Wisconsin. That was at Monsanto and at Agrocetus, a company working under contract at that time in Monsanto. Now, I know it's probably going to sound, wrong to say, but I didn't have anything against Monsanto. They were a company that serviced industrial farming. And they were trying to do something really sensible for industrial farming. They weren't trying to be, a small farmer's friend. Well, a small family farmer in the United States or Canada or something like that. Yeah, sure. But their job was to improve the quality of industrial agriculture, decrease the imprint on the environment, make it profitable, of course. So they spent years figuring out all these different silos they had to organize: intellectual property, marketing, pre-elite germplasm, distribution, troubleshooting, which is critical in a product. All these things they figured out, except the one missing link was they didn't know how to genetically engineer soybean. So they had everything ready. And then what did this moron drop on their desk but the tool they needed to genetically engineer soybean? Which is kind of what I wanted. What I didn't want was it to be a monopoly. So they did the hard yards. They worked in a disciplined way to get ready to make an intervention. They needed a last piece of the jigsaw to pull it all together. I provided that gratis. I didn't understand the world of intellectual property. I certainly didn't sign this form to get it or anything like that. I said, here it is, guys, go. And I discovered something that's to this day most open science advocates don't know. It doesn't work that way. It favors those who can capture it and use it. Those who can't organize and incentivize all the other parts of making a product will just find themselves doing science and publishing it in science. And it won't move the needle one bit in fair play. It won't move it in environmental sustainability. It won't move it in profitability of farming, which is essential. So over the subsequent years, I just realized that my naivete had ultimately participated in increasing the dominance of one multinational in international agriculture for a long time. I mean, I liked the people. I really did. I liked all the people in the private sector with a couple of exceptions, and in the public sector too. They were my friends, but they didn't grok it. They didn't understand that you have to align a lot of different actors with a lot of different cultures. You got to respect them. You got to listen to them. You got to let them find an incentive to participate. Just like, we do with publishing and getting promoted. They have to have incentives too. Sometimes it's financial, but not always. So I realized then that open had enormous potential to disrupt, because at that point, if you watch the change in plant genetic engineering publications, it was hockey stick, elbows up. So it was hockey stick, and everything changed except who benefited. I mean, the scientists benefited just fine, and they went on to get careers and everything else, but it didn't change a thing in the world. And that wasn't what I was interested in. By this time, I got seduced by narrative of truth, of people that mattered, and which wasn't me, it was them. I had a toolkit, and I could make toolkits, but they were the ones who were being disadvantaged by the status quo, not me. So from my perspective, it isn't open if it isn't open for others. It isn't open if it isn't open for outcomes. And from my perspective, the open fetish that we're enduring, which I developed and I worked in, and I ultimately I got past that part of it after the subsequent 30 years of my career, is so omphalosceptic. It's so obsessed with itself that stopping back and say, what's this for? What are you trying to achieve? Are you really thinking this is the bottleneck to improving the well-being or the response to climate change or the ability to galvanize new and different problem solvers who generally aren't scientists? And it's not. In my opinion, it's quite primitive. I think the stage we're in the open world is very, very primitive. But I'm sorry, I've been blah, blah, blah. I don't want to have a sip of wine. So on with you.
So this was about the time of the biological open source initiative. So presumably and I believe that was 2005.
Well, no, actually, all this stuff happened in 1980. I joined the United Nations for a couple of years because I figured that if most of the world was not living in an industrialized world where the sort of blandishments I experienced were norm, I had better understand it, and I better understand international arenas. So I joined the Food and Agriculture Organization, the UN, as their first molecular biologist, which is really a rush to get a laissez-passer with a hologram that says molecular biologist, but it doesn't say what country you're from. You just let me in, I'm the UN molecular biologist. What a rush. And I learned a ton. I learned that international bureaucracies are like other bureaucracies, filled with normal people, some well-intentioned, but captured by their own incentives and their own rewards. It was really wonderful. I spent a lot of time in many places I'd never would have gone before, with the Atomic Energy Agency as well as the FAO and others. And that's when I started Cambia, which is the institution that I still run, whose goal, Cambia, of course, is from the Latin root for change. Its goal was to change the demographics of problem solving. I guess that's how I'd frame it. We have a problem that the demographics of those who can solve problems is constrained to those who benefit from those problems being solved. What a surprise. And those who are early adopters, which is generally rich people like me, or, well, you know what I mean.
Yeah.
So we thought a real point of invention, and this is, again, a methodology, is if we design and distribute broadly tools that enable successes beyond the guild. So building bridges between life scientists and other types of human beings that can tell a narrative if people know how to listen, and we could collectively solve problems. And again, the biological open source started in 87 or 88 when I sent all that stuff out, but I hadn't formalized it as an actual initiative and launched it until 2005. Part of that was that at that time, when I started this, the whole thing of sending out all the DNA and all that and the manifesto of decency and blah, 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 it happened at the same time in a different Cambridge with a different Richard. So at that same time, literally the same year, Richard Stallman at MIT was promoting the open, it was called at that time the Free Software Foundation. A friend of mine, Eben Moglen, was his general counsel. And it was really clever and really driven. I don't think he was driven for, helping other people solve problems. It was a more theoretical, I don't know I can't put myself in his mind, but it didn't seem to have that. It morphed into the open source movement. I mean, what he was promoting wasn't open source at all. It was a very viral technique, a very viral approach to software engineering, where if you touched that software that had the particular kind of license, it was like tag, you're it. And so the next software you built with that had to be distributed the same way. And the real world, of course, happens with a huge requirement that software engineers don't just serve as software engineers, they serve as people. Flight control towers that keep Qantas planes from falling out of the air. Those are not software engineers. They're air traffic controllers. You have to think that software is about solving other people's problems. It really isn't about software. Software is a tool for you and I and everybody else to make faster, better, whatever else decisions. But basically, it's those decisions that burn the economy, burn everything else, that make something happen, right? So this is where it gets really strange. The parallels between software and biotech were not small. They were serious parallels. They were informative parallels. But the truth is, open source as a community phenomenon had one unique strength. The beneficiaries were the innovators. If you're a software engineer, you use what you contribute. I mean, you get a kernel, you get a new build, you use it, you benefit from it. So there's a constructive feedback loop where software engineers participating in open source benefit themselves. Now, if I'm doing open source tuberculosis or open source biotech or open source malaria, it's not because I got malaria and I've got to get fixed. It's a totally different thing. And also the time frame and the cultures required to create benefit are very diverse, which means software engineers don't have to respect anybody to benefit from an open source project. They just have to like their software, and it works. But it doesn't transfer into everything else. Now, at first, I thought, well, software is like code, like DNA, and so therefore we can develop an open source initiative. And we tried. We tried hard. We were very sophisticated by that time about intellectual property. I was very, very skilled at it. I used it to fund the institution with a tiered price model where everyone had access. I would have licenses to Gus from Monsanto, but also from a ma and pa shop that made it was a farmer collective that made the first transgenic papaya that saved the industry in Hawaii. And they did it for the price of a Martin guitar. And so we negotiated licenses, but what can you afford? And there was a little mob in Canada that was doing potato biotechnology, and there were 3 people. And he said, well, how about if we do you some T-shirts? And I said, well, could they be polo shirts? I'm a grown-up now. I want to wear a collar. And so they made, this is powered by biological open source. That was great. And they got a license. And it was kind of cool. But it was a hard lesson because we developed something during the course of the biological open source. There was a new technology that beat around 1,000 patents. So at that time, gene transfer to plants was effectively monopolized by a small number of global multinationals, many of whom had been either subsumed by or cross-licensed with Monsanto. But they weren't the evil empire. They're just doing what businesses do. They were optimizing their ability to deliver to their shareholders and to the community a product.
Mm-hmm. Mm-hmm.
You know, companies are not the bad guy. Monopolies are, which means it's our failure as regulators, as citizens, to ensure that the rules are fair. And they play by the rules generally if the rules are articulated and fair. And I had no trouble with any of the players in ag biotech licensing, knowing that the licensing was going to develop Cambia. Because, why? Because it's a golden goose. You keep developing new technologies, treat them well, you're in an ecosystem. Business works that way. You have outliers that are jerks, but not many, because they have to be there next week. So anyway, what ended up happening was we started this thing. We looked at all the patents. At that time, I had a brilliant head of intellectual property at Cambia, the institute I formed in Australia, with help from the Rockefeller Foundation to troubleshoot their rice programs. And they did it with a nod and a wink because they had nothing in their 5-year plan to support this clown with the idea of setting up this institute. But the genius who was running their rice biotechnology program knew that I was, again, foolish enough to kill myself for this, and he hired me to troubleshoot their whole program all over the world. And we weren't incorporated, so he wrote the first grants to me personally, you know, and I'd never had that much money in a bank account in my life. And then we did such a good job after a year, he wrote 4 times as much money. Oh my God. And he said, you might want to think about incorporating the institution. You know, it's getting really hard to justify writing a check for $500,000 to this guy. And, but he was utterly brilliant in how important it would be to get new methodologies distributed and supported. So he really loved the sort of the biological open source because it was getting people to realize that the world of science is much more than just science. The knowledge that, I mean, if you were to look, let's take an example, Michael, if you were to look at any of the COVID vaccines or any medicine that's important in almost anything, and you look for the companies that produce those, which have enormous R&D budgets, enormous. And you look for their presence in the scholarly peer-reviewed literature, it's basically negligible. You won't find much from, in those days, Monsanto or Novartis or GlaxoSmithKline or, you know, a couple of papers here and there when it was low-hanging fruit and it was okay. Somebody's leaving, they need a goodbye, so they get a paper. But you won't see it. Where you do see it is in the patent literature. Lots and lots and lots of it. And one of the reasons I got pissed off about the open fetishists is that is the largest open access corpus of knowledge on earth. It's not effectively copyrighted, which means full text, 150 million documents about every field of enterprise, including some stupid ones, like scholarship too. But they don't read them. It's a cultural thing. You show me a scientist who's talking about open science, who's bothered to look at the patent literature, which is rich with ideas and inventiveness and dorky stuff. It's not about saying no. It's about saying, this is what I've done. The patent system, Michael, was developed by my great-great-uncle in the United States anyway in 1793. And he was not only invented this patent system, he was the first patent examiner. Imagine the Secretary of State, and who's, you know, Thomas Jefferson, really smart guy. Imagine you have to go in there and show a model of what you've invented and tell him it's new, it's not obvious, and it's really important. You know, the bar of examination was pretty high. He was a smart guy. And Eli Whitney, developer of the cotton gin, had to do exactly that. He had to take a little model of his cotton gin, go in there and convince Thomas Jefferson that it really was worth it. And what the purpose of the patent was, and still could be, if the open fetishists would pay attention, the purpose was teaching. You know, the root of the word patent is patere, which is to lay open. The purpose developed was so that secrets, which were how people proceeded, how people progressed, if they developed something, how could they progress if they just tell everybody? How could they raise capital? How could they do anything? How could they get credit? So if you tell everybody what you know and what you can do, you have a very limited right to be the first person to make use of it in a commercial setting for a while. It's for a while in carefully sculpted, if it's done right, claims. And it is like every industry reads these. Almost no academics outside of a few in engineering and chemistry where it's a part of their culture. It's so frustrating. And so we developed this thing, this way of building what we call landscapes of what is this, the landscape of patents around a particular technology. We read hundreds and hundreds and hundreds of patents and realized that all these gene transfer patents, and this is where it gets really cool, all these gene transfer patents said that this bacterium, Agrobacterium tumefaciens, it's a particular soil bacterium, was shown to transfer DNA in like a Trojan horse into a plant. And then they built all these. There's a method to do that with corn. There's a method to do it with corn on Tuesday with green socks. There's a method to do this. And there's all these different ways of doing it. And some of them really work. But they all claim that one thing. And I thought, well, how can we bust this monopoly? I thought, whoa, what if we convince a different benign bacterium to do it instead? It's not agrobacterium because the claims are really carefully constructed. They say this genus and species does the thing. So we thought, oh, okay, what if we invent a technology where a completely different bacterium can do it to all different kinds of plants, and then we develop a patent-based commons which says anyone can use this if you agree not to rain on the parade of someone else using it for that purpose. If you do other things, you can compete with products, but let this technology be a common core element. And it got cover of The Economist, the New York Times, and paper published February 10th of 2005 in Nature. It was incredibly popular. Really, wow, they're doing all this open source stuff in life sciences. And 2 years later, I shut the whole thing down because it was a complete failure. Biological open source. The idea of it made me very happy. The reality of it made me really discouraged because we had thousands of academics sign on to a site we made called BioForge. And after 2 years of them talking on this site, it was a good forum and stuff, not one had increased the quality of the technology. Not one had contributed an improvement back, which is how open source works in software. You always contribute something back to the kernel. It gets better. It gets better. They had no reason to do this because they had to publish quickly and in a prestigious journal. So they used the old technique that's patented by other people because academics don't know or care about other people's rights. I mean, it's just a terrible structural problem. They don't know how hard it is to make a product. They have no respect for the people who manufacture or the people who regulate. It's just a cloister. It's so frustrating. So ultimately, we realized it was going to be just. I was going to get kind of crass here, but it was going to be really a pathetic activity where we're just maintaining this thing so everybody could talk to themselves and be a virtue signal. So we just shut it down and realized, you know what the problem is? Academics don't know anything about intellectual property. They don't understand how valuable it is to read it. They don't understand how frustrating it is. If you do your work and it's captured by the holders of intellectual property that you don't know, then you don't decide the distribution of your work. When someone says, oh, I'm publishing it open, that's like what I did, stupid moron. Me, you. And it doesn't work that way. Oh, I published it open access. Big deal. If that publication enables only one or two or whoever, private entities or public entities, to practice that in a commercial setting, does the benefit reach the people you need at a price they can afford? And short answer, generally no. And so the problem is we need to mature the open science and open access movements to realize it's not all about you, buddy. It's about other people. So open for others is what matters. That's all that matters to me. And open for outcomes. If it isn't making outcomes that are fixing the environment, fine. I'm not saying it's a bad thing, but it isn't what you think it is. If you're going to sell it as this a transformative thing, get your act together and realize it's only transformative when the narratives go both ways and everybody telling it has respect and engagement in the process. We never did in science. So BioForge and the biological open source morphed into, I put down my pipette, stop being a wet scientist, and realized the answer is going to be in building bridges between the scholarly world and the enterprise world of making stuff, and later on to bridge it into the regulatory world, into the policy world and stuff. So the issue is building bridges, and it's not enough to build it and they will come, because they won't. They'll stay where they are, because that's where they're rewarded. You then expose partnerships that can be cost effective. They can be new opportunities for funding, because that, of course, motivates almost every academic. And they have new opportunities for companies to do things together with academics. So considering what's going on right now and the complete decline of modern American science through unilateral action, and the danger, for instance, that librarians will be asked, this is where I get really passionate, because my mother taught me that a librarian is pretty much, she didn't say it, but I saw they were the heroes of society. And in a world where we're just stunned with fire hoses of misinformation and disinformation and real information, but fire hoses, we have to have guides. It's not about AI, it's about humans, human intentionality, and the best librarians are patient public servants who guide through this miasma of crap and to help you find relevant, actionable knowledge out of the miasma of crap. And so from my point of view, librarians are the greatest heroes in society and precious. And when recently a couple of your colleagues in what's the name of that journal you work with?
Katina.
Katina.
Yeah.
Joel, these two guys, Joel Thornton and Curtis Brunby, just did a marvelous piece of public good sleuthing. They actually used freedom of information to finally bust the confidentiality clause of Scopus and Web and Clarivate, or elsewhere in Clarivate, about databases, old crotchety databases that everybody uses because nobody got fired for buying IBM, nobody got fired for licensing Clarivate. Well, now what's happening? You look at it, and almost all the universities on the list, Canadian and American, they're spending $500,000, $300,000, $400,000, $500,000 for something we now can give them with fully open data for 5% of that, and it's sustainable for us. And so I view this as a personal challenge. This is how we save librarianship. We say we have to throw the administration some bones. Don't kill the don't axe the librarians. Look at what you can do from open community-driven and supported initiatives that can let you cancel databases that are old and tired and non-responsive and closed. And if you do that, you'll be telling your university librarian or the provost, look, we just saved instantly $500,000. Oh, you're doing your job. And parenthetically, we didn't have to fire our research librarians or our digital librarians. It's a real thing. It's real contribution. So when I saw what Joel and Curtis did, I don't know them personally. I'm on LinkedIn buddies, but I don't know them. But wow, that was thoughtful and really clever, because you don't force a university to bust a clause that they've signed on to. You use the freedom of information transparency. Really thoughtful scholarship. Now that was useful. So I'm now probably the next couple of days going to go public with a comparison of what they are paying, these universities, and what we charge to the same universities, which is typically 5%, for something that's based on open data and includes all the global patents and linkages between them. Because the last thing, I notice I'm blowing away your time. I'm so sorry. But one of the things that's really critical about the lens, from day one, it wasn't just about searching or even exploring. It was about understanding the knowledge flows between different cultures, whether it's science, intellectual property, business, manufacturing, regulatory. These are cultures. Every one of them has a vibe. They have a set of norms that they adhere to by and large. Usually the dinner conversations, the wine conversations I have with human beings are uplifting. And as soon as they go back to work, it's kind of predictable what they'll act on. It's based on their norms. So our job has been to shift the normative dialogue between the guilds. So we started with patents because we cut our teeth on the largest non-copyrighted full text corpus there is. So we learned a lot about full text work before anybody else. We launched years before Scopus even existed. And we've been doing this a long time, running a 24/7 infrastructure. And when you cut your teeth on every patent ever existing and all the complexities of patents, which are really, really complicated compared to scholarly works, then when you bring in scholarly works, you've heard of Microsoft Academic Graph, which was developed as a major contribution by Microsoft to their great Microsoft Research, to their great credit. It was driven by a wonderful man, a really good friend of mine named Kwansan Wang, a truly generous spirit. He did something that Google would never do, which is to take it off your ecosystem and say, take it. Now, they were able to do things through crawling that basically we're not 100% sure what the legal status is, but Microsoft has more lawyers than God. So who's going to sue Microsoft for doing some scraping? So they didn't. So they did a real thing. And we, for 5 years, were their only collaborator. Everybody used their stuff, but our teams got in the code base and we uploaded 50 million records into their system. They were great people to work with. And I was really sad that Microsoft reverted to basically saying, no, we've done that. Kwansan left. And I miss that. And another great friend of ours, David Lippman, old friend for 40 years or something, he started NCBI and PubMed, and we worked with him a lot too. And Crossref, really phenomenal mob. So we collaborated with Crossref and actually MRC and PubMed to map for the first time the knowledge flows that existed in a citation graph between patents and scholarly work. So we now have this thing, we call it INFORM, which is the International Industry and Innovation Influence Mapping, as before influence had such a dirty term. Mm-hmm. But the idea is to look at the knowledge that, let's say you or I had published a paper. Imagine I published a paper. And that's cited by patenting entities, which are not evil necessarily. In fact, most are not. They just do something. And so they have to. They're required by law to tell the patent examiner, what did you read that laid the groundwork for this? What did you read that gave you the tools for this? What did you read that proves that your stuff is not obvious? In other words, prior art, enabling art, contextual art. And they say what it's about. It's not like scholarly citations. You know, one of the biggest elephants in the room that we don't talk about citations is most of them are bullshit anyway. I mean, when I do write papers, and I have written papers, plenty, usually it's just in the manuscript, say, gee, you just have to have a reference here. What do you know about, oh, yeah, we'll put such and such. And what happens here? And you go, oh, well, who's going to be on the study panel? Oh, such and such and such. Very few of them are because people have thoughtfully read them and said, oh, this is important to this conclusion. So the people who are marketing AI approaches to understanding context, they say, well, yeah, except that that's not how most scholars work. But we don't talk about that. But this one, they have to do it because it's the law. And so they say, yeah, it's prior art. And even the examiners, what's called the classification examiner or the first principal examiner, will read it and say, hang on a second, I remember seeing something on, and then they put something down as prior art, and then, you know, the applicant is going to have to argue with them, and they generally lose. So what we're doing is we're trying to build this map of knowledge flows. So for any problem, now, I care super passionately about climate change, but not just to carry a placard, to see how can we get people who aren't jerks to be able to make electric cars? How can we make X, Y, and Z that are real contributions? Well, it's not going to happen without all of these different cultures doing their thing. And so we have a visual, Michael, that I think is really helpful. We call it Rainbow Bridge because it's a rainbow bridge. Go figure. Imagine a suspension bridge with maybe 6 or 7 pylons, however many. One is science or research, another might be intellectual property, one might be manufacturing. Then there's regulation, and of course, business skills and civil society, which is a very important amplifier of needs of creating narratives. I mean, because when you got 7, 8 billion people, you can't listen to everybody, so you have representative narratives. So it's not surprising. But when you look at this, you think, wait a minute, there's all these different pylons in a suspension like this. And by this metaphor, you don't really get an outcome for society, which is the pole over on the right, unless it goes through all of this stuff. You can't make a product that doesn't meet all these requirements. You can't change practices without putting new processes in the hands of people who need to change those practices. You can't do it by saying, oh, if only. What you can do is lead them with a better alternative. And to do that, you have to align all these different ones. And here's where science screws up. Actually, everybody screws up this way. The silo I live in, it must be the driver for all this because this is where I am. And so that silo can go way up. More science is required. Wait a minute. I would say more coordination is required to know what the different pieces of the puzzle are. If you're going to actually map the future, not just pat yourself on the back for the past, something that guides every decision you make should be the context. Who really should be brought into this discussion? Who should we listen to before we lift a pipette or an electromagnet? I mean, what should we do as activist scientists where the activism is not, holding placards and supporting some really good politicians? There are a few. It's actually doing stuff with the skill set we have. It means we have to be more generous. We have to be better listeners. We have to care not about more money in our silo. We say, wait a second. So in this particular work, let's say you're doing something on low temperature superconductors, or high temperature low superconductors, like they all are. And you know that it's really important for, let's say, fusion technology. And you know how important that is. Do you know who's doing research in your field that's in the private sector? Do you know who's reading your work? Do you know who you should talk to now that you're not going to get a DOE grant? Them. So we're trying to portray it as a way of making every step one makes from now on more informed about the context, the partnerships, the directions we need to take to deliver to society, which I think is the core beauty of science, which is driven by the core genius of librarians. I mean, the purpose of a librarian, the core purpose of a librarian, in my view, is to do wheat from chaff. To come back to the plant metaphor, there's just so much information. How much of it is actionable knowledge? What's trustworthy? And these days, there's more and more misinformation and disinformation in the fire hose. We don't need more data. We need more humans that are invested in understanding that. And we've got them. They're in libraries. They have this human intentionality and context and drive. I've never met one that wasn't, you scratch them hard and they are passionate about public good. You can't say that about most scientists, but most librarians, you'd be hard-pressed to find one that doesn't care passionately about that. You got to give them the tools, like a job for one thing, and then appreciate them. Let them help. Let them help people find actionable, meaningful, evidence-driven knowledge including knowledge of the cultures of these other silos. So these librarians have a great future ahead of them if they start paying attention to patents, start paying attention to regulation, because they can introduce, as the gabarit, the guides, they can introduce to scientists, to funders, whoever else, here is the panoply of actors that needs to be assembled and coordinated and incentivized if we're going to take, if we're going to take Janine's phenomenal idea about such and such and turn it into a product that actually can work to improve the productivity of small farms. To get that to be done, we need to coordinate this. And of course, the usual response is, it's not my department, says Werner von Braun. I can go up, who knows where I come down. That doesn't work anymore. So that's what the lens is about, and why I stopped being a wet scientist, which I really missed. And you did want to ask one other question. I'm way over time on about the whole genome theory, which is the core of everything I think and believe in. And I'll talk about it another time, I guess, because I'm way over time. But this is critical. We have to understand that when there's a flood of information, there is nothing more important than contextual knowledge that is driven by the public spirit, and that is librarians. So our purpose in the lens is to give them the tools to save a ton of money, participate in a community-driven open product that isn't just about data and being cool and virtue signaling. It's being part of a solution set, because that caters right to their hearts. And I have never met, we had a wonderful woman, Chris Burg, who is the MIT librarian, was on our board of directors for a while. Wow. Quality people like that. You just think, you die and go to heaven, you hope they're like that. And anyway, so that's what we try to do. The whole genome theory, which is the most important science I ever did by a factor of 100, is something I did in the earliest stages of Cambia. But then I had this sort of moral requirement to focus on Cambia. So I put it on its back burner. So right now, I'm in the process of trying to leave Cambia and turn the lens over to a new steward, a new entity that can take it to scale. So I can pivot before I lose my marbles completely on whole genome, on presenting it, on thinking about it and talking to people about it, because it puts the whole microbiome world in a particular context. And whether it's right or not, I don't know, but I think it is. So anyway, I'm really sorry I've just been talking like this. I live out here in the bush in Australia, and I don't have a chance to talk to people very much. So here we are.
Well, it's a great story, Richard, and remarkable. Sort of, you've described enough for sort of for several careers, from geneticist to should we call campaigner to what you term as a social entrepreneur, and enough topics covered in this in this talk to sort of to fill many interviews. So I think we'll have to stop there. But I mean, it's been amazingly it's been fascinating listening to your perspective and your take on not just science, but the way science is done, the way knowledge is disseminated. And I think there's so many topics we could pick up on. So anyway, thanks very much for your input, Richard. It's been great listening to you, and I look forward to maybe we're going to talk to Charles to talk about some your current initiatives on another talk.
Love that a lot. So let's plan on one, okay? And thank you so much for arranging this, Michael. I appreciate it very much.
Thank you.